Hello everyone, and thank you for attending this presentation today. This talk is focusing on how medical CT scanning methodology was used to compare porosity development of two Paleokarst horizons from the Silurian in southern Ontario. Our goal is to demonstrate the many advantages of the methodology developed specifically to work on full diameter rock core. Full diameter cores from the Bruce nuclear site were made available for medical CT scanning at INRS in Quebec City. The core samples come from two water-bearing Paleokarst intervals, the Lower Silurian Ground Formation and the Upper Silurian A1 carbonate unit of the Salina group. Medical CT scanning is a non-destructive approach that can be completed on full diameter cores and provide 3D quantitative data on the size, shape, and distribution of the macroporosity. The next two slides will give you a little bit of context. The study area is located on Lake Huron, and 15 wall core samples were analyzed for the project with core diameter of 75 millimeters and lengths varying from 10 to 55 centimeters. In terms of lithostratigraphy, the upper Silurian karst horizon is located in the uppermost part of the A1 carbonate unit in laminated mudstone. In contrast, the lower Silurian karst rubble is present in the entire thickness of the Guelph Formation. The methodology we used in this project was previously tested on reference core material such as the Berea sandstone or the Indiana limestone, and had proven to efficiently match core average porosity measured in conventional gas Prosymmetry Labs. The principle is simple. Samples are placed in a dry chamber and scanned in a dry state. Subsequently, the chamber is flooded with distilled water and a vacuum is applied for 24 hours, degassing distilled water and samples at the same time. The samples are then maintained fully immersed for up to 78 hours and are scanned in a saturated state. Samples are scanned 3 times in both dry and saturated state, and the scans are later averaged to decrease noise level in images. The dry and saturated scan data can be co-registered and processed using a combination of MATLAB and Aviso operation. Because we deal with the subtraction of the dry and saturated dataset, we can derive effective porosity value without having to assume the density of the rock in the equation. This is actually a key component of this two-stage saturation methodology compared to conventional way to derive porosity from medical CT scan dataset. Here is a summary of different outputs obtained from each one of the wall core samples we analyzed. There is two main porosity aspects that are assessed. First, the porosity is evaluated throughout the sample with an average porosity value comparable to those obtained with gas porosity. But we also provide porosity profile along depth or visualization through multiple cross-section or 3D volume views. In addition, after applying a segmentation technique, statistics on macropore and mesopore are produced, such as an estimation of the pore diameter, distribution along depth, or the pore distribution histogram. Moving on now on the results section of this presentation. On the left-hand side, the figure illustrates the typical results one can get from scanning sample the conventional way in a dry state. In this instance, the porosity is calculated assuming a uniform mineralogy and calcium carbonate, and hence a uniform density through the entire sample. On the right-hand side, the figure illustrates the result of the two-stage saturation methodology. So in black and white, the coronal section illustrates the amount of water that was replacing the airspace after the saturation technique. In order to highlight the advantage of working on full diameter, a regular 1 by 2 inches core plug is placed in the bottom right of the slide. A1 carbonate sample we analyzed have an average matrix porosity along depths of up to 4%. They also often show large rounded to elliptic macropores concentrated in 3 to 4 cm thick beds, which are then associated with much greater porosity value along depths. To gain more information on the macropore, the next important step is to proceed with the segmentation technique, where a threshold is selected to separate the rock matrix from the pore spaces. On the left, there is a coronal section through the connected porosity volume. The areas in white correspond to areas where water replaced the airspace. The different level of gray on this coronal section represent the amount of water that was filling the pore space. As you can see here on this example of an A1 carbonate sample with 3 different thresholding scenarios, the thresholding selection is a crucial step. The threshold selection has to be made in order to realistically capture the macropore network without giving too much weight, unrealistic weight, to macroporous areas. Those macroporous areas are detected by the saturation technique, but they are not fully resolved at the medical scan scale. Once a threshold is selected, statistical data on pore size and pore distribution are available. Here are the results from the same A1 carbonate sample we were looking at. Large pore, 100 cubic millimeter and higher, are restricted to the upper part of the core. They totally disappear in the last 6 cm of the core interval. Small pores are abundant in this sample as well, as you can see on the histogram, but pore reaching 100 cubic millimeter and higher account for almost 50% of the total pore volume. Finally, the video output for this A1 carbonate sample. The video output is a key component to visualize the result of the threshold selection and make sure it's a relevant threshold that was chosen. On the left, the 3D volume represents the pore network created once the threshold selection is made. Each color represents one connected pore space. On the right, the 3D volume is a representation of the same pore network in 3D, but there, the color bar represents the amount of water that was replacing the airspace. The information here can be understood as the porosity intensity. The medical CT scan cannot totally resolve small pores, as we said before, but this is how porous and microporous rock spaces would appear. In other words, the volume on the right-hand side with the color bar express how filled the pore space is, and you can see here on the right upper corner of the slide a thin section photograph illustrating the complexity of pore spaces in A1 carbonate. Now let's go to a representative example from the Lower Silurian Guelph Formation. In Guelph dolostones, the average porosity matrix along depth was found around 5%. Locally, samples display very large raggy macropores causing rapid radiation porosity up to 12%. This is what we can see here on this example. The largest macropores appear to be connected through a network of fracture and macropores unresolved at the medical CT scanner scale. Here is the threshold selection for this Guelph sample. On the left, in the same fashion as for the A1 carbonate, we have a coronal section through the connected porosity volume. On the right, we present 3 different thresholding scenarios for the Guelph sample. And this is another illustration on how critical the section of a threshold would be for further analysis. This is the result we get for the Guelph sample once an adequate threshold is selected. In the Guelph sample, very small pores, 10 cubic millimeter and smaller, are very abundant. But the contribution of very large pore, 500 cubic millimeter and higher, is definitely the major contributor to porosity, representing 96% of the total pore volume. Here is the video output for the Guelph sample. On the left, the 3D volume represents the pore network created once the threshold is selected. Each color represents one connected pore space. On the right, the 3D volume is a representation of the same pore network in 3D, but there the color bar represents the amount of water that was replacing the airspace. The 3D volumes in the guava sample highlight very well the abundance of very small pores. They appear in blue and green on the right-hand side volume. The small macropore create a connected macropores network. The 3D volume also illustrates the location and the geometry of the large localized pore. They appear in red on the volume on the right-hand side as well. The fracture planes are also visible when the 3D volume are rotating, especially in the upper part and the lower part of the sample. A regular 1 by 2 inches core plug is presented in this slide again, and this is to illustrate how the location of the subsampling for conventional gas porosimetry could lead to overestimating or underestimating the porosity depending on the location of this subsample. Now, to conclude, there is several advantages to the medical city analysis we developed and applied to Silurian karst sample for Southern Ontario. First of all, the methodology is non-destructive and can be applied directly on large core samples. Secondly, the two-state saturation protocol and the numerical comparison on wet and dry datasets give information on the connected porosity. The methodology is also a powerful 3D visualization, giving insight into the pore connectivity in 3D. After segmentation, we have access to statistical data on pore and pore network. There is two main limitations to the methodology. First of all, the resolution of the medical CT scan is limited. As we mentioned several times, the medical CT scan resolution does not allow to resolve the macroporosity, even though we do have access to information about the location and the contribution of the macroporosity. The other limitation is the thresholding step in itself. It is a very crucial step, but it could be a tricky one. To actually help with the thresholding selection, we are running petrographic and SEM studies at the moment. So stay tuned and look for our next paper on the subject.